Hello, my name is Alexis Morrissey, and I'm a PhD student at Penn State University. And today I'm going to be talking about my method Allo, which can be used to rescue multi-map reads in ChIP-seq and ATAC-seq analysis pipelines. So getting into some definitions and motivation, the most important definition is what is a multi-map read? It's a read that aligns to multiple locations in the genome with equally high alignment scores. And so how does this rise in ChIP-seq data? This happens when a transcription factor binds to a repetitive region of the genome, and therefore any reads that are within this repetitive region will now become multi-map because this repeats in multiple locations in the genome. And the reason we care about this is because most standard ChIP-seq pipelines actually discard multi-map reads, and that means that we're losing a lot of data. And even more so when you look at this graph here. So between 12 and 30% of the genome is not uniquely mappable with the 35 to 75 base pair reads that we use in most ChIP-seq data sets. What this graph is showing here is this is looking at over 100 different ENCODE data sets and examining how many multi-map reads on average there are in those data sets. And the average was almost 16%, meaning we're throwing away 16% of our data set whenever we do this. So what are in these regions and why do we care? There are a lot of studies coming out that are showing that transposable elements are involved in gene regulation and they affect a lot of biological processes. And so some of these that have been discovered previously is they're involved in mammalian pregnancy development, interferon response, pluripotency in various cancers. What this graph is showing here is it came from a paper that was looking at 26 different orthologs in human and mouse and looking at the overlap between transcription factor peaks and transposable elements. And as you can see, there's quite a high overlap and the average was about 20%, suggesting that a lot of transcription factors are actually binding to transposable elements. So there are current methods that do deal with multi-map reads, and all of them rely on the uniquely mapped read counts to do their probabilistic mapping. So here's just like a little toy example where you have one multi-map read that maps to two locations. This location has 7 reads, this location has 1. And so the probability that a read would belong to that location is 7/8 in this case, and 1/8 in that case. So now I want to get into our method Allo and how it's slightly different from these previous methods. So Allo actually uses 2 different metrics to decide if a multi-map read belongs in a location. The first way it does this is it does exactly what I previously mentioned, where it takes the total uniquely mapped read count around multi-map reads. But the second thing it does is it looks at the distribution of those uniquely mapped reads. So in ChIP-seq data and related sequencing, we have peaks, and peaks do have a characteristic shape. So we thought we might be able to exploit this and able to deal with this problem, as peaks would be more likely to receive more reads and thus more multi-map reads. So what we did is we trained a neural network to do binary classification on peak versus not peak. And we did this actually using images because CNNs are really good at predicting on images. And so essentially how Allo works is it looks at each multi-map read individually, in each location it could belong to, in each location it looks at the total uniquely mapped reads there, it looks at their distribution, and it makes a decision on where a multi-map read belongs based off of those 2 things. Another really important thing about Allo is that it has very simple pipeline integration. So you just take your sequencing reads, your FASTQ file, you can put it in bowtie or BWA, you just have to add 1 extra argument. Out of that, you'll get an MMR inclusive alignment file. You put that into Allo, whether it's a SAM or a BAM, and then Allo will output a SAM or a BAM file for you, which can go into any p-caller and can be used for any downstream analyses. So it's very easy to integrate into pipelines that people already have. Since this is a computational conference, I think it's important to talk about the CNN training and how we actually generated our training set. How we did this is we took ENCODE data sets. We had 2 different versions of each data set. So the first version is just using the regular pipeline where we have the uniquely mapped reads that are full length. So this was 100 base pairs in this case. Then we took those uniquely mapped full length reads and we actually artificially shortened them. So some of them actually became multi-map reads, but we had a ground truth for them. And so using the full length UMRs, we then called peaks using max2. So now we have true locations for peaks. And in some of those locations, some of the reads became multi-mapped reads after being artificially shortened. So now we have a UMR distribution at peaks that contain MMRs, and we know this should have been a peak based off of the full-length data set. And so in total, this then became our positive data set. We took these read counts, we turned them into pictures, and that was our positive data set. Our negative data set was very simple, and the way we created that is we just took regions that had multi-map reads but were not called as peaks, so just background regions, and that became the negative set. All in all, Allo was trained, has two different neural networks, and so the first one was trained on narrow peaks and the second was trained on mixed peaks. The narrow peak data sets, there were 10 of them, and they were all transcription factor data sets, and the binary classification was 94%. For the mixed peak data sets, we had 9 histone modifications, and the binary classification was about the same. It was almost 94%. And to choose between using the narrow or the mixed peak data set is just one simple argument that you give Allo. So getting into the results, it's very important for us to compare Allo to other current methods. And so the way that we created a data set to be able to do this is we did kind of something similar to what I had previously mentioned. So we have full-length reads here. We artificially shortened them, and then some of them became multi-mapped reads after being artificially shortened, but we still have their true location, the ground truth. And so what I'm showing here with this read allocation accuracy is I'm saying if a read belonged to chromosome 1, position 100, did it go to that location? And as you can see here, Allo outcompeted random and read count only. And so read count only is literally just using the uniquely mapped read counts and not the neural network. And read count only is actually very similar to the method MUM rescue light. So it could be like a good proxy for the difference in accuracy there. But regardless of narrow peak or broad peak, Allo was able to outcompete these two methods. The next thing we looked at is we wanted to compare Allo with the newest method available for multi-map read allocation, which is SmartMap. The problem with SmartMap is it outputs a bed graph file, meaning we don't have the resolution to do this type of one-on-one comparison. So the best thing we can do is we looked at percent error of average read depth within the peaks. So if a peak was supposed to have an average read depth of 10, how close was it to that based off of our ground truth? And so as you can see here, Allo outcompeted SmartMap because it had a lower percent error or a similar percent error in every single data set that we looked at here. One thing to note is it seems, at least in the narrow peak data sets, which would be these right here, that Allo seemed to do better when the data set had less noise. So we're showing FRIP score here, which is fraction of reads in peaks. CDCF and ATAC-seq had a higher FRIP score, and Allo outcompeted SmartMap quite a lot in these two data sets, but not so much in these 3 Krabs finger data sets. This didn't seem to have as much of an impact, though, in the histone modifications, but it is something to note that Allo and SmartMap are quite similar in noisier data sets, or at least from the ones that we've looked at so far. The next thing we wanted to do is we wanted to do an application of Allo. So what we did is we took 200 different ENCODE data sets and we analyzed them not just using the uniquely mapped reads and then using Allo to see, how many more reads or how many more peaks are we getting? What's the percent increase? And so the mean percent increase was actually 13.5%, meaning we're getting 13.5% more peaks. And that's a lot of data that would have otherwise been thrown out that may actually be important. Another thing we wanted to look at is we expected the Allo peaks to have significantly more overlap with repetitive elements than the uniquely mapped peaks, so the peaks found even if you hadn't used Allo. And so there is a slight shift towards this. And so the pink or reddish color here is uniquely mapped, and the bluish color here is Allo. And like I said, there's sort of a shift here. So there is slightly more repetitive element overlap, but not as much as we expected. Another thing to note is these are very high fraction overlaps. And the reason that this happens is because we used repeat masker annotations, and repeat masker is quite liberal in its annotations of repetitive elements. So it isn't surprising that a lot of uniquely mapped peaks are also overlapping. But like I said, we did expect the shift to be greater. So the next question we thought to ourselves is, what is causing this? Why isn't there a huge shift where a lot of these peaks, these allo peaks are actually in repetitive elements. So we actually discovered is a lot of allo peaks are also coming from gene duplications, so paralogs and paralogous families. And so this is a histogram showing exactly that. So when we overlapped allo peaks with gene duplications, we actually found that 20% of all allo peaks were overlapping with gene duplications. So that could add into the reason why not all allo peaks were overlapping with repetitive elements. Some of them are actually overlapping with paralogs. And so one, really cool example of this is the example of BRCA1 in the Gage family. This was something that hasn't been seen or mentioned previously in the literature. So the Gage family is part of the cancer testes antigen group of genes, and they've been shown to be upregulated in a lot of different cancers. So it wasn't really surprising that this interaction was there, but it's never been actually shown before in the literature. And so here is the before and after, basically, of these peaks. So using UMR only, this is what the peak looked like. It wasn't called a peak in that data set, but after using Allo, you can see that the peak is very visible and has high resolution there. This is another example in Gage 2A. This is uniquely mapped read-only, and then this is using Allo, and you can see you gain a lot of resolution. So in total, Allo identified BRCA binding in 50% of all the genes in the Gage family, which is very interesting. So some big takeaways is number one, we developed a software that rescues multi-map reads and ChIP-seq data, and it's easy to put in any current pipeline. Number two, outside of transposable elements, it appears, at least to us, that duplicated genes are a significant source of multi-map reads and ChIP-seq data. And number three is don't throw away your multi-map reads, don't throw away this data, because you're getting rid of a lot of biological significance when you do that. Allo is functioning and it's available on GitHub to use. Please email me or contact me with any questions, or if you think that there should be an extra argument Allo, because I actually really take these things into consideration. For acknowledgements, I just want to thank my entire lab. I also want to thank Penn State and especially the Center for Eukaryotic Gene Regulation. And within that, especially the Russ Hardison lab for all of their great suggestions and support, and also using Allo in all of its worst, beta states. And so thank you for your time.